Hi, my name is Charles Warden, and I have one cat named Bastu. Bastu has various genomics data collected for her. The blog post in the background describes whole genome sequencing data for 2 samples submitted to Basepaws. This YouTube video focuses on a subset of the blog post content related to a C to T color point mutation in the Basepaws reports. So briefly, I've tried to upload everything that I can. The raw data that I have available right now is in a different format for the second sample than the first sample, but I will update GitHub as I'm able to create reads that are in a more similar FASTQ file format for the paired end reads. What's perhaps relevant right now is that there are PDF reports for 2 reports. They're both labeled as version 4.0 reports, but one of them has a correction, the CT variant that I'm going to describe here. And just very briefly show that if you click here, it will load the report. Hopefully it will load the report. And you can scroll down. If not, you can click download, and that's another way you can view anything that's up here. And what I'm trying to say is that I have specific notes and code uploaded for reanalysis, but essentially I have everything that I could upload, I have uploaded. So for example, VGL's the UC Davis Veterinary Genetics Laboratory, that's our report from there. You can see there's a lot of stuff from Basepaws, PetQ-Check, Wisdom Panel, and so on. So if being able to see any of these results is helpful for you, I hope the GitHub link is something that's useful to know about. In terms of this YouTube video, Basepaws has a blog post where Bastu and I are acknowledged. So I think this can be described as saying that these cats have the Siamese Colorpoint Restriction trait, and this is a picture of Bastu. So Bastu does not have this trait, but there was an earlier version of the version 4.0 Basepaws report that said she had 2 copies of a variant that caused this trait, and that she was likely to have the trait. However, I was able to work with Basepaws and show that she does not have the trait, and explain in terms of her own raw data from Basepaws that she should have 0 copies instead of 2 copies. And I believe this is important because there were other cats reports where they were originally saying that they had 2 copies and are likely to have trait that they did not actually have. And my understanding is this should be fixed for other cats. But hopefully if I go through what I did, then maybe others could go through similar things in other situations, and Basepaws would also appreciate and acknowledge them as well. So in terms of the earlier version 4.0 report, there's this Siamese coat color trait, and there's this C to T variant, and it says that Bastu is supposed to have 2 copies. And when you do have 2 copies, you are in fact likely to have the trait. The problem is that she has 0 copies instead of 2 copies, but that's what I'm going to walk through in greater detail. So here's a screenshot. Now the earlier report's a little bit harder to see because it's smaller, but essentially in June 2022, you can see there's a red box. That's where it said 2 copies incorrectly. So she was likely to have the Siamese Colorpoint trait. But then as in August, which is after the blog post and after the discussions that I'm trying to basically summarize, you can see there's a green box. The green box is representing that says 0 copies instead of 2 copies, and she's not likely to have the trait, which better matches what she looks like. Now, distinguishing these 2 is kind of hard if they both say version 4.0, and for whatever reason you only had a PDF. But at least in terms of what I can see for Bastu's whole genome sequencing samples, I'm not 100% sure if it's also true for the read plus health results, but I would guess it could be. And there's an extra trait. So if you look at the summary page for both version 4.0 reports for Bastu, then they both say she has 1 likely to have trait, but the trait changed because an extra trait was added. And so this is basically how I can currently tell the difference between the earlier version 4.0 report and the newer version 4.0 report. And I'm going to follow up with Basepaws, see if I can get any information, and I'll add a comment if I do have anything else I can say about that. Either way, here's a picture of Bastu again. Again, Bastu does not have the color point trait. She's a tortoiseshell, and she has color shown throughout her body. And there's this figure that describes what the color point restriction trait is. So essentially, I might describe this as a more restricted color towards the middle of the body and a better ability to show color in the cooler parts of the body, like the tail, the paws, the face, or the ears. And by talking to Basepaws, I could figure out that this C to T variant is the exact same Siamese Colorpoint Restriction variant that's indicated here by the UC Davis VGL, and it's this C superscript S. There's, I guess, more than one way in which you could have two variants that cause the trait, but I believe it's called Siamese Colorpoint Restriction because it was first discovered in a Siamese cat, and a lot of people, when you think of Siamese cats, you perhaps think of this trait. So if you know all the possible variants that cause this trait, then you can say that you should not have the trait if you have 0 copies or 1 copy. And so Bastu is like this wild type cat, and it says capital C underscore. So the capital C is for the wild type variant, the C in Basepaws notation, and the UC Davis is using this underscore notation to indicate that the cat can have either 0 or 1 copy. If the cat has 1 copy, I don't believe you can tell that from looking at the cat, but you can know that to basically, if for some reason it's relevant to know whether the cat could produce kittens with the trait if you had 2 carriers and then they could produce the trait. And so here is a screenshot of the cat ancestry results. Even though it's called cat ancestry, it has some trait or phenotypic markers. And here's capital C, capital C. So this is the way the UC Davis VGL describes it. And the Basepaws terminology, it would be 0 copies. And this is basically 0 copies for any of the markers is what capital C means. And for individual markers, the 0, 1, or 2 copies. So this is by Basepaws. For Wisdom Panel, they also check the same exact variant, and they just describe it as color point discovered in the Siamese, and they use a wording somewhat similar to Basepaws, and they say that she should have 0 copies. So at this point, we have 2 results from 2 companies or organizations, and they are different for the same variant than Bastu's from Basepaws, at least for that earlier version 4.0 report. Now, to be fair, it's possible that what's commonly said is not true. So it is possible that you could have 2 companies or organizations say one thing, 1 company or organization say something else, and the 1 company could be right. However, in this particular situation, we have access to Bastu's raw data, and by reanalyzing the raw data, I could come to the conclusion that the result from the other 2 companies or organizations was the correct interpretation, at least for this particular variant. So there's a little bit of a complication here in terms of figuring out where to look. I won't say too much about this, but I will say that I was able to start doing this from a position provided by Wisdom Panel for what I might call the canonical or perhaps common or standard chromosome D1. And that's the correct sequence to use for Felis catus 6 or Felis catus 8. But as of the recording of this video, Felis catus 9 is the most recent. But as I understand it, you have to look at 2 different sequences to get the functional version of the gene. So I'm going to describe alignments to this particular position because this is what's a better fit for Bastu's sequence. And if you looked at alignments to that position, I believe there was no alignments to that position. And that's why I went back to the earlier versions and realized that this other, what people call supplemental chromosome or can call supplemental chromosome, is more similar to the earlier builds, but just the name supplemental chromosome can be a bit misleading. We're not actually expecting it's a separate chromosome. It's just the assembly can have separate sequences, and usually you think of those sequences as being chromosomes. And so here is an alignment to Felis catus 9 at that alternative position. And so the idea is that when scientists work together to put together a reference genome that we hope is relatively similar to most cats, the reference genome sequence is down here at the bottom, and the variant position is right here. Now, this is a G, and if you remember the Basepaws report, it says it's a C to a T variant, so that could cause confusion. The thing that's important to understand is that this is DNA sequence, and DNA is double-stranded, so you could actually represent the sequence with either of the strands, and it just so happens that the genome build uses this strand, and the reverse complement of a G is a C. So if you use the other strand, it is a C. Now, it's hard to see, and I apologize about that, but there's an A in this read here. However, there are no A's at this particular position we want to look at, and the reverse complement of an A is a T. So if you were to see a C to T variant, you would look for A's in the reads, but there are no A's in the reads. So that's consistent with Bastu having 0 copies instead of 2 copies. There's only one alignment shown here, And as I mentioned before, that actually I have to do some reformatting to be able to use the same scripts for her second sample. And it was a little while before I received that data, but essentially if I'm trying to describe what Basepaws I believe is acknowledged before, that I didn't have that data available at that time, so that's why I'm just focusing on the first whole genome sequencing data for the sake of trying to describe that discussion. That said, one thing that I learned I did not expect is that even though I purchased whole genome sequencing data, and I did get whole genome sequencing data, the report, at least as of the earlier version 4.0 report, is based upon Amplicon-Seq data, which is what I believe the breed and health results are, or more specifically, the breed results, I believe, are coming from imputations or guesses about particular positions in the cat genome for low coverage whole genome sequencing data, and these Amplicon-Seq data is higher coverage at certain regions for the health and trait markers. And so this is a trait marker, and for those other kits it would be used in the Amplicon-Seq. And even though you could have made use for whole genome sequencing, in this case it looks like Basepaws did the other sequencing, and that's what they used for the report. So what that means for this discussion is that I can use two different data types for one cat, and I'm going to show that they both would show that she has 0 copies instead of 2 copies. So now the top part of this view in Integrative Genomics Viewer, also called IGV, is the whole genome sequencing data from the screenshot. It looks a little bit different because the way that whole genome sequencing data works is you break the DNA into small fragments that you can sequence with Illumina sequencing, and with these paired in libraries, you sequence from each end. And so, for example, this is a fragment up here. So this red or pink color is a read. Then you have a gray line where there's no sequence, and then you have this blue or purple read. And they're different colors for on different strands, because going from one direction, you'd go from one strand, and then going the other direction, you would have to start on the other strand. And here's a bigger fragment where, again, see, I'm just going to call it the red read, a longer gray line, and then the blue read. And then here we have a smaller fragment where you largely sequence the same sequence on both reads. So you have red, then you have blue with a gray line inside it, and then you have the blue without the gray line. Now the amplicon is somewhat like this fragment where you have primers for the ends of the sequence, and you can see that down here. This is the amplicon, and it's called an amplicon because you have those primers to amplify a particular sequence. And the sequence is sufficiently large that I believe they used a program for their own custom code to merge those reads out of a fragment that looks kind of like this. And they provide me intermediate form of data. So they did not provide me with the same type of raw data that I have for my whole genome sequencing, but I think it was still useful. So what I did is I took the text file of that intermediate data, I reformatted it, and I tried to process it in a way that's similar to the whole genome sequencing data. And there's only one color down here, and a possibility is they have a strand-specific library so that next to the primaries, you have the adapter sequence that could be directly sequenced, or it could just simply be that they wanted to make it easier to see the sequences. So a double-stranded, a non-strand specific library where you could in fact check variants against each strand against the reference genome existed, but they only, this intermediate form data is all set to the same orientation. So that's why there's all pink. But essentially, to make sure you understand the difference between those library types, if your genome is if your cat's genome is a relatively good match for the reference genome, you should get somewhat even coverage across all positions in the genome. And then Amplicon-Seq, you should get high coverage where you've designed those primers, and you should get little or no sequence elsewhere in this particular view. You see it's no sequence, no gray bar here or here. I did not sufficiently well explain the difference between the 0 copies or 2 copies conceptually. I can explain at the end, because I think something happened in library preparation such that the end of the amplicon is not the biological sequence. And I could say that in terms of, here's the whole genome sequencing data. We can see there's no variants of any type. It's all the reference genome. And then you can see this gray bar turns into a red bar, and that is IGV's, I guess you might say, informal variant call, or it made some decision there was likely to be a variant there. And in each of the reads, there's a T. And so the T color is red. That's why this is red. If it was like half red and half green, that'd be like you had T and A, and the reference was a C. So this is the difference between what 0 copies or 2 copies look like. However, please note that we look at the position that it took a little bit of extra effort to find, but is a relatively good match for the previous genome builds, is you can see there's no A's up here for the whole genome sequencing data, and there's also no A's down here for the Amplicon-Seq data. So with this reprocessed data, that reprocessed using open source programs, which means free programs available to the genomics community and anyone who has the ability to run the code and do the analysis, then you'd come to the conclusion with the reanalysis of raw data that she had 0 copies instead of 2 copies. And indeed, the most recent Basepaws reports have made that change. And I believe that is what they would like to acknowledge me for. Now, if you don't have the ability to do that and visualize the data after you reanalyze it, there's another option. It's not ideal if you don't have the whole genome sequencing data, but let's say you discovered a discordance between what your cat looks like and what the report says, and you wanted to try to have some amount of discussion. Basepaws was willing to be kind enough to provide you with an intermediate form of data like they did for me. And to be clear, I only have it for this one Amplicon. They don't give it to me for the other Amplicons. They just gave it to the one where I was reporting a problem. So what I did is I took a text file, I imported it to Excel, and I sorted in alphabetical order, which is for this second column or column B. So sequences that are identical to each other are now next to each other. And I use a formula in Excel, which I can write here, which checks 2 cells to see if they have the same content. So in this case, cell B6 has a sequence that's a different length than cell B7, so therefore they should not be the same. It should say false. And indeed, that is what it says. You can drag down the formula, and it just so happens these 2 sequences are identical, and these sequences, which are different lengths, must also not be identical, and it says false. So I've done this work for you already, and then wherever it says true, you can go over and up and know that those 2 sequences are the same sequence. And you had a large set of trues next to each other. I highlighted it in yellow. So if I scroll up and down, then you can kind of visually see this sort of thing is jumping out. This sequence is jumping out. If you have a cat that has 1 copy of the gene of each of her, or his or her, 2 chromosomes, then if you have one sequence that appears, 100% identical sequence that appears more than anything else, that would mean it really should be 0 or 2 copies. And when you look at this, you might think it is more confusing than the whole genome alignment or even that reprocess alignment. And it is a bit more confusing, but as far as Amplicon-Seq data goes, I think it is reasonably good. And I think you should always expect some amount of off-target amplification. And for whatever reason, it's not unusual to have sequences that are not the biological sequence in the amplicons. And I think the difference of the nucleotide at the end is a not so bad example of that. Something else could make it even harder. But if you assume that the cat can have variation in this region, the primers still amplify what it needs to amplify, then having one sequence that is 100% identical, that jumps out at frequencies greater than anything else that's 100% identical, we can copy this sequence using Control-C, and then I am going to use UCSC BLAT. I'm going to paste the sequence here using Control-V. And if this is the first time you've used it, then it will probably say human. But if that's the case, you can just select the species, pull down, and select cat. As of the time of making this video, the latest genome build is Felis catus 9, and that is what I've used to provide a position to check for this variant. So we're going to click submit, and it's good. It says that position 1 to 176, which is what matches her whole genome sequencing data, it's 100% matched to the genome build. That last nucleotide is different, but that last nucleotide is not different in your whole genome sequencing data, so we're not going to focus much on that. So at this point, because it's 100% identical, and we know the reference build as the wild type sequence, then you know she has 0 copies. However, if we're going to imagine some theoretical cat where maybe there was some complication or something, so Basepaws is willing to do this for some different customer, I'm going to manually change a nucleotide. and I'm pretty sure this is not the nucleotide where I want to look, but it says 176, and now it's not 100% identical. But that doesn't mean your theoretical cat has 2 copies of the variant of interest. And the reason I selected this is so that you can visually look into this to try to figure out whether your cat has 0 copies or 2 copies. Again, making the assumption the amplicon amplifies the sequence of interest like it's supposed to, even if there's variation in the region, and that you basically had some way that you could pick out representative sequences relatively good by eye. So relatively well by eye. So if I click browser, this is the reason I use this is because the BLAT is associated with the UCSC genome browser, and so your cat sequence, this theoretical cat sequence, which is 1 nucleotide different than Bastu sequence, is shown here, and the part that I change is this red part right here. However, when we go to this table and we copy and paste the position that is for this particular variant, it has the red spot is not red here. So essentially, this is the reference sequence. It's a G, like we saw before, and you just need to remember that Basepaws uses the reverse complement, so that's the C, and there's no sequence here, so there's no A or anything different than the reference genome. If we zoom out, assuming it's not the whole amplicon, but covers the sequence I changed, you can say I did make a G to A change. So if this G to A change was at the position that you're looking for, then this theoretical cat would have the variant. And hopefully that could show you how you could have something that's not 100% match, and you could figure out whether that is the part of the amplicon that is a known variant that's been previously characterized or not. So I hope this has been helpful and useful. Essentially, I walked through the details of exactly how you could use raw data from Basepaws to come to the conclusion that she should have 0 copies to 2 copies, and the newer reports now say she should have 0 copies instead of 2 copies. And so if the underlying concept is useful in other situations, I hope Basepaws gives you a similar acknowledgement as they did to me. As a sort of closing comment, I'd like to point out that there is a changelog at the bottom of this blog post. So I've never made a YouTube video like this, but I assumed it would be harder to make changes for the YouTube video than the blog post, and so I've waited longer to create the YouTube video. However, the idea of the changelog is supposed to be similar to the automatic history on GitHub, where I posted the reports and code to analyze raw data for Basepaws, and any raw data, even though it's usually from Basepaws. So I think this is something that's important for transparency. So even though I want the version I think considered would be the best, the easiest to find, I want to make sure I also am clear about when I make changes and what changes I make. Now at the very bottom, there's a place where you can add comments. And I certainly welcome any questions, comments, or concerns. You can also do this on YouTube, but I think hyperlinks might get flagged. So if you did, there are certain things where it might be better to share stuff on blog posts rather than YouTube, but either way, I will do my best to provide responses if you do have feedback. However, please remember that I am not an employee of Basepaws. I am a bioinformatics specialist, but I do general genomics research, and I won't know the answer to all possible questions related to your cat's genetics or genomics. Also, you will probably be receiving responses outside of normal working hours for that reason. Nevertheless, I really do want to do the best I can to help, and so if you have anything that's important and you think there's something I can do to help, please let me know, and I will try my best. I hope you found this interesting and useful. I hope everything goes well with you and your cat or cats. Thank you again. Bye.